Welcome back to Acing AP Biology. In this video, we will be introducing the process of DNA transcription. In Unit 10, we defined genes as a unit of inheritance with a specific location on chromosomes that generate phenotypic effects. Genes can also be defined as a specific nucleotide sequence in a particular location of the genome. The end product of that gene's expression can either be RNA or a specific protein. The genetic information in DNA exists as a specific sequence of nucleotides, which are transcribed and translated into proteins. These proteins impart specific characteristics or phenotypes to the organism. Transcription and translation are the two central processes mediating the pathway from gene to protein. The sequence of nucleotides in DNA determines the primary structure of protein, which is the linear sequence of amino acids. The path from gene to protein, therefore, goes from DNA to RNA to protein. DNA is transcribed into mRNA in the nucleus. mRNA is then further modified before it leaves the nucleus. The mRNA, which is holding the transcript for protein synthesis, leaves the nucleus through a nuclear pore and attaches itself to a ribosome, which is a protein manufacturing organelle. Ribosomes are composed of rRNA and protein. Transfer RNA, or tRNA, is also involved with translation. The mRNA sequence is then translated into a linear sequence of amino acids, which is the primary structure of a polypeptide. The protein eventually folds into its three-dimensional native conformation based on the initial amino acid sequence. For a reminder on protein folding, visit my macromolecules video from Unit 3. In prokaryotes, there is no nucleus, so DNA is not isolated from ribosomes, allowing for translation to take place immediately after transcription. However, in eukaryotes, the intermediate molecule mRNA is required to take the genetic information from DNA to the ribosomes outside the nucleus. Although the process is slower in eukaryotes, RNA processing can take place due to the spatial segregation of DNA and ribosomes. There are 20 different amino acids. Triplet sets of nucleotides code for amino acids. So for every 3 nucleotides, 1 amino acid corresponds to it. 61 out of 64 nucleotide triplets code for amino acids. The other 3 code for stop signals, which we will discuss a bit later. These nucleotide triplets are called codons. mRNA codons code for the amino acid sequence during translation. The anticodon on tRNA helps tRNA stay in place, binding to the complementary mRNA sequence while translation takes place. It is important to remember that in RNA, uracil replaces thymine, so mRNA codons will always have a U, not a T. Let's say the DNA sequence is TTC. The mRNA codon would be AAG. The anticodon on the tRNA would be UUC. The amino acid that pairs with codon AAG is lysine. The start signal for translation is codon AUG, and it codes for amino acid methionine. The 3 stop codons that code for termination signals in translation are UAA, UAG, and UGA. These code for a release factor, not an amino acid. That's it for this lesson. In the next video, we will go over the process of DNA transcription. See you then. For more bio content, hit subscribe.